Welcome to our presentation on online science labs biology. I'd like to present our two presenters, introduce them to you, Suzanne Wakim and Tanya Hoerer. Suzanne Wakim is coordinator for student learning outcomes, distance education, and open educational resources at Butte Community College. She is also a biology instructor who has developed numerous online courses, and she has created online educational resources for Carnegie Mellon, OpenStax, Nature, Discovery Education, and others. Dr. Hoerer teaches diversity of organisms, introductory biology, ecology, marine science, and human anatomy at Coastline College. She works with internship students on research projects in the fields of marine biology, ecology, and human anatomy. She earned a PhD in biological sciences from Syracuse University, where she studied microbial ecology and ecosystem sustainability in Yellowstone National Park. I got all that out. All right, we have decided to let Suzanne start, as she will be presenting resources that biology instructors might be able to use in a science online class. After that, Tanya will take over and give a tour of her class as she discusses more pedagogy issues. So let me hand it off to Suzanne and you can begin.
Great, thank you. Let me just share my screen and hopefully you can see that. And if you want to follow along, just because I know it's nice to be seeing slides on your own, and there's a bunch of clickable links. So if you want to go to the slide deck, you're welcome to follow along there. So as Bob mentioned, I'm going to be starting us off with a framework of how to build science labs online. And then I'll walk through a list of resources and tools that you can use to help you build your online labs. And then Tanya will share a specific example of how she has done this for her class. We did want to start with just a quick introduction to the challenges and advantages that we face in our online labs. And this, I think, is where a lot of us are coming from, is we want this framework of where are the challenges, and then in the presentation and in future discussions, we can take a look at how we can address those challenges. And the advantages are a nice reminder of why we are making this effort. So over on the right, the advantages are mainly that it allows access to more students who have more complicated lives. You know, they have families and jobs and whatnot, and may not be able to attend our face-to-face labs. And of course, in the current situation, this applies to everybody across the board, because none of us can attend our current face-to-face labs. So more access is 100% access at this point. It also allows, one of the other advantages is it allows students to work more slowly and at their own pace, right? So we've all had those students in our labs where the 3-hour block is not quite enough for them, and they benefit from being able to slow the pace down to whatever works best for them. And the last advantage, I think, is the one that's most exciting to me personally, is that it really brings biology into the students' lives and into their home and into their space. So they start seeing themselves as biologists rather than, you know, you go to a certain place over here, which is where biology happens, and then I go home, which is where biology does not happen. So those are some of the advantages. The challenges over on the left side are that the hands-on component can be difficult, right? And that's something that we'll address in a little bit, but students don't have these great tools at home, the microscopes and the, you know, all of these models and whatever. So that can be a challenge. You also want to make sure to verify student identity and ensure academic honesty. And so we'll look at some strategies for doing that. And you want to make sure that the at-home labs have the same level of rigor as our face-to-face labs. So like I said, we'll address many of these topics as we go along. The framework that I wanted to present is what's called backwards design, and this is actually a framework that is for any course design, online face-to-face, lab lecture, but I think it works particularly well with labs because labs are fairly unitary, right? So each individual lab is its own entity. And so we can work through this framework for each lab to help us address those particular needs. So the framework is you begin with what are the SLOs for that lab, right? And the SLOs are in our course outline of record, and there's things we all love and embrace universally, I'm sure. But they are also our agreement with the students and with the transfer institutions and employers and everybody else. This is what our class is for. Like, this is what our class is doing, right, the SLO. So you always want to start with that. Then the process of backwards design is to step back and say, well, how do I know students can do that thing, right? How will I assess their ability to do whatever the SLO is? And then finally, at the end, you take a look at how can I build activities that help them demonstrate their ability to do the SLO. So a quick example of one SLO, and this one I got from, I think, one of the CID courses. It's a pretty common one: identify and describe biological molecules. And actually, I should pause here because, this was in the description, but it bears repeating. The focus of our presentation today is going to be looking at non-majors general education biology. I already saw in the chat there was a question about majors. We'll have opportunity to discuss those at the end in the Q&A section. But for the core of the presentation, we're going to look at non-majors just as a place to start. Okay, so this is a pretty common SLO for our non-majors. If we think about how we can assess that, there's a few things I might want students to be able to do with biological molecules. I could really focus on having them classify them. So if I show them a chemical structure, do they understand what that is and can they name it? And if that's the assessment, then the lab activity down here could be some form of 3D virtual simulation, which is great because then they can visually see the 3D models. I could have lab kits, right? So the atomic model building kits, which is nice and hands-on. Or I could do a DIY, at home building models with marshmallows and toothpicks that we've done before. So there's lots of ways to get students to see and visually experience those 3D models. If the goal of my class is more about having students understand the lab tests, right? So this is how you test for sugars, this is how you test for proteins, then I may want to lean a little bit more towards the lab kit or the kitchen chemistry to do that lab test. Or I may want to lean towards nutritional analysis and have students go out and look at how are foods determined and how are the nutritional values of foods determined. So different sort of approaches. Again, this can be more hands-on, or I can lean towards more application. And it's really all about how do I want students to interact with that information. And then there's one more example there as well. So that's just an overview of how I've approached moving classes from face-to-face to online. And the rest of my section is going to be walking through different places that you can find resources to help you build your online lab. And if you're following along on the Google Slides, all of these links will be clickable. And you may want to click them later if you're inclined to get distracted and focus on that. So a good place to start, I think, is thinking about which labs absolutely must have a hands-on component, right? So if you list all of your labs and the SLOs for each one of them, are there some labs that just really need that hands-on component? And in that case, you may want to have students purchase a lab kit or have them purchase materials for kind of a DIY kitchen chemistry type lab kit. The next slide here has a list of all the places that I found lab kits, and there may be more, but this is a good place to start. The advantages of lab kits, right, is that they feel real, right? They're really hands-on. They bring that hands-on aspect directly to the student. The other advantage is that they're fairly straightforward on the instructor end because they come with the lab instructions and they come with the scaffolding of helping students understand how to interact with this material. So a lot of the work has been done, which is nice. The disadvantage is these can be really expensive depending on how many of these you require of students and which kits are students being asked to purchase. And so you may want to think about which labs really need the kits and which labs maybe don't need the kits and bring that cost down a bit. I know some schools for the current situation are looking at CARES funding to help purchase these kits, which is a great short-term solution, but not, I think, sustainable in the long term. But the key here is I think that not all labs will need the kits, and so just limit to the ones that do. The other disadvantages are that supply chain issues, which I saw in the chat there, that right now, because so many of us are moving to kits, some of the companies are having a long lead time. And so that's something to look into sooner rather than later if you want to go to the kits. Some of the companies have a very long backlog. And then the last kind of thing to keep in mind is that students who join the class at the very last minute may not get their kits in time. And so it's good to build in a strategy for that. Okay, so that's the kits. Oh, and one of the questions that tends to come up often is what about safety, right? So is it, if students hurt themselves, what about liability? And a lot of the kit companies will have safety contracts and insurance and stuff built into them. So that's a nice advantage to the kits over the build it yourself. But I think in a lot of cases, it's just easier to have students build it themselves. So like, for example, here, these beads, I assume, are for the mitosis, meiosis lab. And we can have students find similar tools in their home. They probably don't have to purchase those. Whereas the frog, we probably, if we needed that, wouldn't want them to go out and catch frogs. So that's one option. Like I said, you can take a look here. There's different companies that'll do that. And the DIY is just a reminder that we can have it built ourselves as well. One company I wanted to bring kind of special attention to with the kits option is Odigia, which is a company that's built off of the OpenStax textbook. So if you're using that textbook, it ties in directly to that. And if you're not, OpenStax has textbooks for GE biology as well as majors, anatomy, micro, and physiology. And what they do is they have contracts with Carolina, eScience, and I think a few others where you can combine the kits, some kitchen chemistry and simulations all into a single package so that students are only going to one place to get the full collection of labs, right? So the simulation lab will be in the same place as the kitchen chemistry lab as the kit. So that's just one example for bringing everything into a single place. And this one, the cost, just so you know, it's $30 for the baseline framework, and then based on how many labs you add into that, students get charged per lab as well. So that's that. Are there any questions so far that I'm missing?
Suzanne, I see one inquiring about a micro lab. Have you heard of any micro lab kits that can be used at home?
I have not. I do see here though, Odigia does have micro labs. I haven't looked into those just because my focus is GE, but it'd be worth looking at those and seeing what they have. And then, yeah, Judith, with the micro, the issue is going to be safety and disposal and all of this sort of thing. Oh, and Susan has micro labs, so we'll get some information in the chat about potential sources for micro labs. That's fabulous. I love it when this crowdsources as well. Great. All right. Other questions? Are we good?
I see someone raised her hand. We can offer audio questioning at the end, the last 15 minutes. So
okay.
If you want to wait for that, you can. But if you want your question asked now, please put it in the chat.
Awesome. Thank you. Okay, so that's the hands-on components. And as I was mentioning, I think a good way to approach this is to balance hands-on with some more virtual or simulations, just in part because of cost, but also in part, I think, because some of our labs are really more focused on data collection and analysis, and those just lend themselves to simulations anyways. I think of even our face-to-face labs, our microevolution section has always been something that's more or less a simulation. So those sorts of things just lend themselves directly to more virtual labs. So I wanted to share a couple of options for virtual labs. And again, there's a slide that has a link to a bunch of them. But this one here is, it's called Lab Exchange, and I don't have stock in any of these companies, so I'm just kind of sharing them as options. But what's particularly nice about Lab Exchange, I think, is the interface is clean and it helps students really see how these tools would work, and it allows them to work with some high-tech tools that we couldn't afford in our labs. Right? So that's one of the advantages of the virtual labs, is it really lets students look at resources that we wouldn't be able to afford, and it focuses them more on the data collecting and the analysis, and a lot of these will have graphing programs built in, and so they can do a lot more of that aspect of science. This one here is also nice in that it's free. The disadvantage of some of the virtual labs is that there's not a lot of chance to run amok, right? So one of the ways that students learn is by making mistakes, and that free-form way of doing labs is a really important part of allowing students different mistakes so that they can learn those lessons. And so when you're selecting virtual labs, that's something to look for. Some of them are pretty, I don't know, kind of click and drag, and I haven't listed those under virtual labs. Those I usually call interactives. The virtual labs tend to have more open-ended options, so I can pretty much click on any of these things and move them in different places and make all sorts of mistakes that the coders may not have thought of. So that's the advantage to some of these tools here. Okay. This is just another virtual lab, so there's a few of them. The reason I wanted to include this one, this is HHMI, is the interface is just a little bit older, right? But what's nice about this one is it has the information right there on the page, and so students are really guided through a really robust learning activity, as opposed to sometimes we've seen students that are just looking through the microscope or pretending to look through the microscope because that's what their neighbors are doing, but they really don't know why they're doing that. This will really help them understand each step, what is the purpose, because the information is right there. So there's an advantage to that. So those are virtual labs. Separated out from virtual labs are simulations. And the only reason I've separated them is I like to categorize things. That's, I think, what makes me a biologist, put things into groups. And so simulations are really more about things. Well, so this one here is some sort of predation simulation, right? And so I add predators and see how the graph changes. And so it's less of a kind of a pretend lab situation, but more of a simulation of external experiences. So these are also particularly helpful for labs like population ecology, evolution, and some of these other labs that are focused much more on data collecting and analysis. This one here, SimBio, does have a cost. There are other simulations like this one here, which is Virtual Biology Labs, which are free, but you can see the main difference is this is maybe not as slick looking, but as far as learning, this provides a really nice way for students to look at island ecology, change the variables, and see how that affects the organisms. So those are simulations, and I am moving really fast, so I'm going to take a breath and see, are there any questions so far?
Yeah, let me back up. Someone asked about a microscope and at a distance, what's the best way to give students a microscope experience?
Yeah, that's a really great question. And there's, I think, a couple of options. One is, and this is where I think backwards design really helps, is the question you want to start with is not necessarily how do we get students a microscope, but what is the goal of them using a microscope? And if the goal is to get a sense of scale, then Stuart, I think in the chat, mentioned a really great solution, which is hands-on labs and virtual microscopes, right? So virtual microscopes, and there's a few linked at the very end, I'll show you, help give that kind of, this is what it would look like. If part of the goal is to have a hands-on experience, you certainly could like loan out or rent out microscopes. Now those are expensive, and there's always a chance for breakage. So that's, if the hands-on component is utterly essential and they need that big microscope, then those are the options, but there's downsides there. The other option that I've been really excited about for my GE class, and this is more for general ed, is the little clip-on microscope that you put on your phone. And before, because I can just feel everybody's eye roll, so just hang with me for a second. The thing I like about it is it's in their pocket, and again, this is bringing biology to them. And so having them just have this little tool, and they're like 30, 40 bucks. They're not expensive. And of course, you're not going to look at microbes with it, but it gives them a way to really look at their world, their space, their backyard in a way that is helping them think like a biologist. So 3 different options: virtual, loaner microscopes, and pocket microscopes, if you will. And then I've only got a few minutes, so I'm going to fly through these. There's a few more resources here, and like I said, you have the slides, so you can click through this. This slide of various resources are places that have lots of really great, but just different collections of different kinds of things. The Open Science Laboratory is out of Britain, I think, and it's part of they have this online, free online university where you can go take classes in anything you want. It's kind of like what MIT did, but it's more global for them. And so there's lots of really cool activities. This one is a skull that you can rotate and measure. There's different kinds of skull. It's essentially an evolution activity. This is a virtual microscope, right? This is one of those I was showing there, where you can look at different plants and see the plant structures versus water drought abilities and so on. And then the very last one that I think we could really benefit from is having students do some citizen science. So there's all sorts of ways for students to actually get their hands into doing science, like for real science, and that is citizen science. iNaturalist is just a site where you find things that are in your area, and that helps folks track what plants and animals are where. And so this is them actually doing real science. And there's all sorts of places where they can do that. Much this is ecology. Protein folding is just a fun, if you've never played Fold It, great time. So there's that. And then if you're interested in having more discussion, because I just flew through a ton of resources, and if you're interested in having a discussion with other biologists across the state, CBC built a really cool community of practice for us within Canvas. If you'd like to join, there's discussions, there'll be some resources and things in there. Please email Bob and he can put you in this community of practice, and then we can work together across the state to build some really cool stuff. Okay, thank you.
Some other questions we can wrap up here quickly. Somebody noted Flash is not accessible. And as far as I know, that is true almost entirely. Anything that uses a Flash player is generally not accessible. Some of these will be Flash-based, some will not. You'll just have to check. I don't think, Suzanne, you went through every single one to check that, did you?
So Flash is I didn't check accessibility, because that just takes too much time, but I did rule out anything that was Flash-based. So that one, there shouldn't be any Flash-based things in here.
Yeah. I don't think Flash is going away at the end of the year. Where it will disappear is using Chrome. Chrome is no longer going to support Flash starting January 1st next year. So it'll be even less accessible. But some of these organizations are migrating these Flash objects to HTML5, which in theory could be accessible. So keep checking back on those. Somebody asked again about molecular biology labs, and maybe that's a place where we can share in the community of practice what people are doing for that specifically. Do students need a computer, or can they work with this lab simulations over a smartphone? Generally, smartphones are not conducive for this. There's usually not enough real estate for a simulation. This isn't always the case. Some simulations can run on a mobile device, but I have found mostly. Suzanne, what did you find? Anything on that particular?
Yeah, I agree with you. These are probably going to be too heavy for a smartphone, but a Chromebook or a smaller tablet type thing would probably be efficient. Right.
And so, yes, it brings up a question of equity. Labster came up issue with that as well. So again, as far as accessibility and equity is concerned, we do the best we can to make the applications we choose equitable and still do the job we need to do. And then where they do not meet our students' needs, we have to find some sort of accommodation plan to help them get access to this instruction. And yes, it's not always easy. Let me look here. Any other questions? Great. Thank you for the sharing, by the way. Not only questions, but great resources people are sharing in chat. That's wonderful. All right, let us now move on, and we'll allow Tanya, Dr. Hoerer, to make her presentation begin, and she will give a tour of her course and talk about how to conduct labs at a distance and student interaction and feedback and all that good stuff. Tanya?
Hello, everybody. I'm going to share kind of a, sorry, I'm getting all my clicks in a row. I'm going to share the course that we've been offering for several years now, our combined biology lecture and lab class. And this is a Bio 100, so this is an introductory to biology lecture and lab course. I prefer to teach the two together rather than have the lecture separate from the lab, but that is an option that we do offer our students. The lecture with the lab is much more cohesive. There's less places for students to click around, so I gravitate towards that one, but certainly what we've done is we've put this up as a model for the department, and so other instructors can separate resources as they feel appropriate. So to begin, I just want to briefly identify the objective that we're going to focus on through this example, but again, encourage you to share and ask questions once I've shared this course regarding upper level courses, majors bio courses, and some different approaches that we've taken in order to work with the precautions we're all taking for COVID-19. So this particular example was designed with two main goals. One is to make this completely online, 100%. Completed through the students' computers, and as a college we do require students to have computers. We do have a Chromebook loaning program to help balance out the equity there for that situation. But students know when they take an online course, it's certainly a science course with us, they need a computer and they need a webcam, because we'll be using a proctoral program that requires them to show their identification. So our intro bio is fully online, which means that we have to meet some requirements through different accrediting institutions, including state and Department of Education oversight. And then our other goal was to create as many open education resources that have zero cost for our biology courses as possible. And so this was one of the ones that we've done with completely no cost. So that meant that we steered away from the kits. I think the kits are fantastic. I think the kits are key to accomplishing the SLOs that need to be met for the course outline of records for our upper level division courses. But for this one, we were able to meet all of our needs by doing everything using OERs. There's a lot out there. There is a bit of a wrench thrown in the works with Flash going away and with making sure that we have exercises that meet our accessibility requirements. So I'm currently actually updating this course, Bio 100, in order to meet some of those requirements. So I've switched out a few activities and I'll share those with you. In fact, one of them is a microscopy lab. So let me just, I'm a very visual learner, so I like to have some of these things visible as I'm discussing them. So before I get into our examples, I just want to reiterate kind of the starting point. And Suzanne did talk about this. When we, especially as we've been trying to navigate the last semester and the next two foreseeable semesters, we've really had to focus very closely on what our course outline of record really allows us to do with these courses. And we've actually gone back to our curriculum committee in order to revise some things in order to offer, for example, human anatomy in a distance learning format, to amend some of the current majors bio and microbiology and physiology courses so that we can have more of a hybrid approach. And of course, to open up the opportunity to offer several of these different courses as fully online. So we had to work with our department, we had to work with our college, so that could be a starting point in terms of how you are able to deliver these courses in various formats. So once we have our format approved, we then have to make sure we can meet our SLOs. So I've included the SLOs for the lab for Bio 100 that our curriculum committee decided upon, and of course is vetted through the CSUs and the UCs. So it's important then to focus on, again, what our SLO is, and then what we're allowed to do in order to assess whether the student has met that SLO or not. And so, of course, this first outcome is one of the best ones and one of the easiest ways to make your online course really a legitimate examination of scientific concepts, and more importantly, the scientific method. And we really simplified this SLO because we felt like this was one of the most valuable tools that our students could walk away with and apply in their everyday life. So as I've been improving some of the exercises, I've really started to walk the students through the steps of applying the scientific method. So I'm going to really focus on this first SLO. These other ones, identifying organisms in the biological kingdoms and addressing evolution. I also try to work on those topics as an integrated approach. So for example, you'll see in the microscopy lab, the first lab, we start to look at some differences at the cellular level of our major, not only kingdoms, but of course domains. And so we work on that as we look at different specimens. And that's kind of linked in with some of these other processes. Evolution is, again, something that I feel is best taught as an integrated concept. It's fascinating to see how many students are comfortable with the cell because they've seen it since kindergarten, but when it comes to evolution, it's a completely foreign topic to them. So again, I try to integrate as much of this as possible. And so the different components of the online lab that I'm going to share with you are designed to include things to make sure that we are meeting our requirements for regular and substantive interaction. We abbreviate that as RSI. From the perspective of Department of Education, they come in and they review as well as our accrediting institutions. At the state level, you might see regular effective contact, right, or REC. And so you'll see throughout the modules, we use discussion forums. This is probably my favorite approach to meeting RSI because the students are engaged with each other. They're required to make a post about something that's biology in the real world, something that affects them. Such as DNA editing, or right now, gosh, we have so many fantastic topics related to COVID-19 that gives us an opportunity to also look at the scientific method, evaluate our peer source, our media and peer-reviewed sources, identify some questions, understand what the methods are, and this is a really engaging activity, I think. So in a lot of my online courses, especially my lecture courses, I offer this as a weekly activity, and that checks the box for our weekly check-in with each other and our weekly interactions, not only between student and faculty member, but also between students themselves. And then there's many different kinds of activities that you can provide feedback with, and you try to do that within the week, right? So we have pre-lab questions. I have this in the format of an announcement that comes out at the beginning of the module that has a linked video. So they're not just reading the information, there's a visual component to that. Of course, you have to make sure that your videos are closed captioned in the appropriate accessible fashion. That means that it's going to have all of the punctuation and correct spelling so that can be read with the reader. Of course, our labs are something that we can also provide feedback with, and this is obviously for a lab course essential. And so to also meet accreditation requirements, we have to make sure that we have a lab manual available. And so you can purchase, there's lab manuals, of course, that are available for the students to purchase. There are lab manuals that come with kits. And then there are usually lab exercises that come with a lot of the different open education resources. What we did is we actually started by writing our own lab manual for our face-to-face introductory biology labs. And then we converted that into an online format. So we took the material, we found interactive online virtual simulations or kind of virtual lab settings. And we modified the goals for the different labs in our lab manuals to fit those exercises. And so those can be either distributed throughout your modules as your lab activities, or you can put them all into one PDF and link that at the end. Exams and quizzes are, I think, the least fun, but they help to kind of check in and have the students evaluate. They're a lot easier to report the SLOs for. Of course, SLOs can be can be linked to some of these other assignments as well. And in order, since we are sticking with the zero cost, in order to really have the students have a hands-on experience, we have them set up an independent research project. And it's very simple. The focus is really on getting the students to, one, understand the steps of the method, and two, really be able to differentiate between what their question is, what their hypothesis is, and what evidence they have to either reject or accept their hypotheses. And so as we kind of train them through the different lab exercises in our online lab course to understand what the different components are of the scientific method, they culminate then by not only accomplishing this, but writing this up, and they have pictures of themselves that they post as they're accomplishing their assignment. And you can just see a lot of pride, a lot of excitement, and it's a really engaging way to meet many different objectives in a science lab that's being done in this format. So that's one of our really fun, kind of integrated throughout the semester. And then we culminate, we have the students look for, I meant to edit that, it should be biology in the news. We have them look for a biological topic in the media. It doesn't have to be peer reviewed, but I have them identify if they're starting from a social media or a news posting. I have them go to the peer reviewed sources so they can identify who actually did the research and briefly explain how the research was done. One of the toughest parts of this is for the student to identify what that research question is, and then see how the methods are designed to address those questions using the scientific method. So we culminate, this is the last assignment due along with their final exam. And so what I've really worked on doing more in the course that we offer now is training them really on that scientific method as we go. So let me show you some examples in the course. The reason I started, let me go back here to the dashboard. The reason I started at the dashboard was because I wanted you to see that this is OpenStax that we use for our textbook. And so the chapters of OpenStax are found throughout our modules. As a practice at Coastline, we like to have a Start Here button that leads them to the orientation module. And then we do like to have this page to avoid so much scrolling through the module list so that the students can get to their content for the week. I'm going to go to module view so I can kind of show you more of an overview of how the course is laid out. So I always start with an orientation module. Of course, in your 8-week courses, this flies by really quick. Even in the 16-week courses, I want the students to read the syllabus, take a couple of quick quizzes, one on the syllabus, one on scientific terms in general. I want to get my welcome out to them. I want them to see all of their resources, learn a little bit about me, identify what types of assignments and policies you're going to have to adhere to, And then also make sure they're clear on how we are proctoring their assignments where appropriate. So we have a little page. We use Proctorio. I have Proctorio then linked to this little learning contract that they're going to sign. They have to show identification of themselves. And with this tutorial included, it's really helped calm a lot of the concerns that students have about using that program. In fact, I think more and more schools are using it, so students are becoming more familiar with it. And then I have them introduce themselves. So I have them do all of this within the first 4 to 5 days, and then we launch into our material. I still keep a very easy kind of overview and introduction for our first module, so that they're starting to get oriented to where to find things in each module, such as their reading, kind of what the layout will be, where we always have an overview of the tasks, things to do for this module, what the objectives are. This is always a homework assignment that they complete on their own. They read, they answer questions. And then again, I have their readings linked throughout each module, and then a little summary that transitions to the next module. These two I keep really light. I'm remembering that students are adding, dropping, they're gonna have technical difficulties in an 8-week course. Again, this is gonna be a lot for them to accomplish in the first 7 days. So again, we're kind of just orienting them to biology and the course in general. After that point, they receive weekly, they receive weekly announcements the whole time, but after module 1, I have little quizzes linked to the weekly assignments. So the idea is that when they get this announcement, they can click to a link on the announcement, or they can, when they're ready, go into their module, and they have to go through these pages of the module in order. So they're going to have to look at their overview task list. They're going to have to look at their objectives, and then they can start their first assessment. This is again linked with an announcement that gives some academic content. That's another way to reach, meet RSI requirements, is by sending out academic content in your announcements. And so this is their first exposure to the scientific process. They watch a little video, and then they're going to take a proctored quiz on the video itself. So I write out what they should be taking notes on as they're watching each of these little videos. So we have one of those designed to introduce each module. Then they move on to their readings and their homeworks. Again, I delay the first lab. Remember, this is a lecture and a lab course, so I delay the first lab until module 3, which means they've had a little bit of chemistry, they've had an overview of biology, they know how to operate their website, they know what's kind of going to be regularly due every week, and so now we get into a little bit more intense concepts. Already they're going to sign up, they're going to choose their project, and so I'm going to give them feedback directly very early on, and I'm going to really narrow down how to properly ask a question that you can actually answer, and that's really where that first focus begins in terms of teaching them the scientific process. So for our first lab, I have an answer sheet always linked that they'll find, and they'll put answers, and then I have all of the instructions and kind of the components of the lab on each page. I think it gives them another option. I think it makes the content feel more thorough rather than just having everything linked to a Word document. So the way I was able to work around Flash this time, and it was tough, I used a Flash Interactive, and it wasn't fantastic, but you could move objectives, you could fine adjust and course adjust. What I've done instead is, as Suzanne pointed out, I just wanted to make sure, one, they know what a microscope is and what the different components are. So I have them start a self-guided tour. They give a really nice overview of a lot of the specifics, but in a very tangible way. And then you'll notice their first set of instructions after they worked through that tutorial that was presented by That particular one is the North Carolina Community College Bio Network, I believe. So they're going to answer some overview questions. We're going to review some topics about cells and do a little assessment. And then they're going to go to a virtual microscope. So they're not going to move the objectives and do the fine tune. Instead, what they're going to do is they're going to see what is visible when you place a sample under a microscope lens. So let me just show you an example. They're going to look at radiolarians. So now they can kind of skip some of the steps that would hold them up in the lab as we're all rushing around trying to teach them how to find their things. And they're going to be able to start looking at the world around them in from a different point of view. So then we also start to introduce different types of organisms, prokaryotic organisms, eukaryotic organisms, single-celled eukaryotic organisms, multicellular eukaryotic organisms. And so I have them also look at some of these smaller components. We have a smaller component of a larger scale, and we have questions that have them think about, where does pollen come from? So we're starting to look at one kingdom of living organisms, plants, right, and how they're reproducing. And then we're looking at these tricky guys, our protists, right, which they're going to have to do a little bit of research and figure out what a protist is, what a radiolarian is, where does it fit in that classification. And already I'm starting to teach them, okay, where is your reference coming from? You do a little bit of research, you tell me what type of a source this is. And then they work into some multicellular organisms with tissues, and Canvas didn't want to have the little image that is linked. I'll show you in the worksheet. But I give them kind of a guide with which they can start to analyze what they're seeing in their image of the cross section of the, I believe it was a sunflower. And they're going to try to map these different components of this multicellular organism onto the image that they've seen. So they start to work with some analysis, and they start to evaluate their resources, their references, as they get some exposure to, again, the view of the world that we use the microscope to obtain. And then I'm just going to give you one other example. So that was their first lab. Their next lab, I give them just the content for cellular respiration. It's a pretty heavy concept, especially for non-majors. And then I have them apply some knowledge for both cellular respiration and photosynthesis with their second lab, which is photosynthesis and productivity. So what we look at, so again, we're starting to introduce some ecological ideas. We're starting to introduce some ideas of different levels of exchange of energy and how that occurs in organisms. And then we also give them a more detailed look at the processes of cellular respiration and photosynthesis. So first they do a little overview. I used again, OER Commons, Open Education Commons. So, and remember, they also got a video with an announcement with a little kind of pre-lab or pre-unit question and answer that I gave them direct feedback in. And so if you notice, I also have started to work them through the steps of the scientific process. So this is their background, which again, they started at the beginning of the module with video linked content with their reading in their chapter. Now they've got another little tutorial that they can use to answer their background questions. And so now we're going to do an experiment to determine how variables, specifically light and temperature, are going to affect the rate of photosynthesis. So they're going to now develop a hypothesis. I'm giving them their null, and I want them, I tell them that I want them to manipulate one variable at a time. And in the analysis part, I want them to look at where they have evidence. Do they have evidence for any of these? And what does that mean in terms of their interpretation? So I found this guy is a very, very simple little manipulation. They can run their sample at different levels of our two variables. We keep things as control in each of the experiments. I have them do it for 30 seconds. They even get a little output of their data there. So if they walk away and they come back, they can see an accurate count. They're identifying their control in each experiment. They're manipulating each variable independently. They're recording their data. And then I have them walk through their data. Now we go to a real world example. What I want them to do is move from this process of photosynthesis that they have been able to visualize the effect of both temperature and light intensity on. And I want them to think about our planet and where we have high light and where we have warm temperatures. And so I give them some background on energy exchange, why we're moving energy through photosynthesis and then through cellular respiration. I'm reminding them to compare the two equations so they can see they're actually the opposite of each other, and they can see the what should be an equal exchange between heterotrophs and autotrophs that gets thrown off balance with the issues that we're seeing now with the burning of fossil fuels. So I have them think about things that we're concerned about in the real world now, things that they're hearing about in the media, and I'm directing them to the living places where we have the highest productivity that, guess what, also correlates with what they found in their experiment. More light intensity, warmer temperatures can lead to increased productivity. And so then we start to apply that to, okay, where's carbon going? And these guys are actually carbon storage sites for us. So as we continue with these types of labs, they'll continue to use this layout. So they're really accomplishing, they're walking through the steps of their scientific method, with each activity. And again, the activities are super, super simple, right? It's just this little guy, but they're able to see how they tested a couple of concepts one at a time, and then comparing the combined results, and then linking that idea from their results to what is happening in the real world and what we are trying to focus on as humans living in the real world. Okay, I don't want to take any more time because we need to give you guys more time for questions. So I'm just going to open it up now for any additional questions.
Tanya, can you hear me?
Yes.
Oh, very good. So let me go back and pull some of these questions out. A lot of the questions had to do with accessibility. For instance, how are you handling the accessibility of labeled images? Do you have alt text for images that you want students to label? And then the flip side of that is, if it's in an assessment and you label it, are you giving away the answer?
So my assessments, I actually avoid labeling in. So on their quizzes, they're either going to be open-ended, so they're writing out short answers, or it's a multiple choice assessment. For the lab documents themselves, I have to have alt text with each image, yes. And we do have resources with the college that can help me do those things. So I lean on my resources at our college level.
Coastline. So if you have a blind student, I'm not sure you have yet, but if you do, and it comes to the assessment where they need to label things and see an image, which they can't do, do you have an accommodation plan ready, or how would you handle that?
Right. So again, I defer to my special programs people and my accessibility gurus to help me make sure that I have that format in a manner that they can see.
And I see a lot of people saying the same thing in the chat. How long do you think the pre-labs take them time-wise, and do they also read a lab manual?
So they have readings in their textbook. So again, this is merged. So they're doing readings for lecture. So I can tie in some pre-lab that way, right? This is to kind of reinforce what they're learning in their lecture topics. For the pre-labs, again, that's contained in that first part 1 of each of those assignments. So that's where they're going to look at an overview. They're going to answer some questions. Most of those, again, are short answer, open-ended, and that's their pre-lab. And then they go into the second part, which is more the manipulation. The pre-lab's really brief. The material leading up to the pre-lab is, again, text reading and in video content as well.
Okay. And by the way, the general guidelines for transferability are pretty general. They leave lots of elbow room for local curriculum committees to design these courses and DE addendums as they see fit. But one thing they do require is a separate lab manual you'll read. But I asked some folks at the Academic Senate statewide, could that take a digital form? And the answer is yes. Could it be broken up into modules and put in Canvas course? And the answer is yes. So as long as it does what a lab manual should do, then you're good to go. What else here? There's some people asking for web links, like to the PS simulation and the URL for the virtual respiration experiment. Can these be provided to the folks?
So let's see.
Maybe later in a document later, perhaps.
So you've got the bio network is already up there. There are some of them that I could not use because a lot of them still use Flash. So I do remember the PS. There was a PS one out. There were some through some of the publishers that they released for free as well, and all of those use Flash. So I've stayed away from those.
Okay.
In anticipation. But I believe those are all provided in the resource links. Yeah.
Okay. And does the worksheet file have all the questions we're seeing that you showed?
Mm-hmm. Exactly. Right.
Very good. What do you find is the best format for student submissions? Word documents, handwritten, photographs? How do they submit their work?
I leave it open to them. As long as they can get it in there, I'll take just about anything. I do have, usually Word and PDF are my preferences. But if a student needs to write it out and they can take a picture with their phone and it's, legible, I'm happy to take images of their documents as well.
Okay. I'm seeing folks asking for specific links to specific resources they saw. And if it's not in Suzanne's document, we'll get that to you later.
Yeah.
So how long did it take to develop the modular format that you showed today?
Okay, so years. We actually had a template that we agreed upon as a school and we were trained on. So that overview, kind of the pre-organization section, the content section, and the wrap-up were already outlined in the template that we used as a college. So I can import that template into any of my shells. I found pretty quickly that I actually started to move away from that in a lot of my other courses, and I just made sure that they could see the overview, the content, and then whatever the activity was, and always a wrap-up. The other thing I wanted to make sure is that there was always a way to touch base at the beginning of each module that had some type of a visual. So it's either me talking out of doors, showing them something, or another instructor. We've pulled resources there, or I found a YouTube that's closed caption that shows, some really neat cellular processes animated, for example. So we always start off that way. We have them evaluate themselves in that first visual exposure. Then we send them into the reading and then the more applied activities. And so, so that's kind of how the process, again, how long I've spent a lot of time, and every semester I update things and redo things. And so there tends to be a lot more front-loaded work with developing any online course. So before your course starts, it's a heck of a lot of work. But then it tends to be, I think, less work throughout the semester compared to what you would do for a face-to-face. So I think it ends up being kind of flipped. So it's just the work's condensed at different times.
Yeah. And then an iterative process every term, making adjustments, getting a little better, a little better.
Yeah,
Okay.
Flash has been a real pain because now I pretty much will be redoing a lot of our assignments.
All right, I'm sorry to say we're out of time. So what I'm going to do is collect all the questions I see unanswered in the chat, and I'll share them with Tanya and Suzanne. And I want to invite everyone attending, if you're not already in our community of practice for online science labs offered by the CBC OEI, I would like to enroll you in that. It's really a Canvas course designed for this purpose, and many science educators are in there already. And we'll create a separate module and a discussion forum just for biology. And we can post those questions along with the answers that Tanya and Suzanne present. So to get in that, I need to enroll you. So send me a request to be in our community of practice for science. Give me send it from your college email address, because I need that to enroll you in this course. And then we'll put you in there and you can get the answers to these questions that we have yet to answer, because there were so many. This is probably the largest webinar/meeting in Zoom I've attended. Thank you. Thank you, Tanya. Thank you, Suzanne. Thank you all for attending. And one little tip, if you like what's in the chat, open up the the chat pane, and you'll see on the bottom right, there are 3 horizontal dots, and you can click on that and save chat. Sometimes they automatically come up when you get out of a meeting, but just to be safe, you can do that to save the chat, and I'll give you a few seconds before I close out, but I will stop the recording